Hi everyone, I'm Gaia. I'm a PhD student at KU Leuven, and I'm also working within the FIBROTARGET consortium. Now we will move on to the in vivo part, so I will show you the animal models that we can use for fibrosis induction and monitoring. So as you can imagine, based on the main research interest that we have, we can use different in vivo models to mimic fibrosis. So let me just, perfect. So the main models that we can use are the chemical induced. So we will have dextran sulfate sodium, so DSS induced colitis, as well as TNBS colitis. Basically, these two chemicals will induce disruption of the epithelium and thus trigger an immune response. In the case of DSS, we will have mainly an innate immune response, while in the TNBS, we will have mostly of an adaptive immune response. Then we will have peroxynitrate induced colitis, which can be interested to study the role of nitric oxide in the context of IBD. Then we will have cell-mediated colitis, which are mainly T-cell transfer induced. And then we can use different genetic strain based, of course, again, on our interest. So we will have strain either knockout for anti-inflammatory gene or overexpressing pro-inflammatory or pro-fibrotic genes. And this strain can basically develop spontaneous colitis or ileitis. Then we will have infection-induced models and some other models such as radiation-induced or post-operative induced. So today I will mainly focus on the models DSS and TNBS induced, and then I will also present you a model that we are using in our lab, which is the TNFΔARE mice. So first of all, I will start with how we can induce the disease, and I will start with the DSS model. So again, as I said before, DSS is a chemical that can disrupt basically the tight junction of the epithelium and thus basically induce a more loose intestinal epithelium, which will then trigger the inflammatory response. We can administer DSS in the drinking water that we give to the mice. And we have basically two setups of DSS-induced colitis. We have the acute model where we only perform one cycle of DSS followed by a recovery period. And this is mostly interesting for the study of early phases of inflammation, so very acute inflammation. While for us, the most interesting for the fibrosis development and fibrosis studies is the chronic setup, where we perform 3 cycles of growing concentration of DSS, and this is due to the fact that generally mice tend to develop some kind of tolerance towards DSS, and then basically by the end of the 2nd cycle and beginning of the 3rd cycle, we will have the fibrosis development. There are multiple studies that showed how DSS can induce fibrosis, and this was confirmed both histologically, where we can see the thickening of the wall of the gut in the mice exposed to DSS compared to the control ones. And again, also with endoscopy, we could see how we have an increase in the granularity of the mucosal surface, a thickening of the colon, and the fibrin became more visible. There are also studies that shows how the stromal compartment can be affected by DSS, both in terms of cell number of different subset of fibroblasts and also from a transcriptomic point of view, both in the case of acute models of DSS and in the chronic model of DSS. If you're interested to do more details on this paper, you can find the citation here below in the slides. Then we will have a TNBS model, which is also fibrotic, so a chronic model that lasts 56 days, so more or less the same as DSS, where we need 63 days to have a full 3 cycles. In this case, we give the chemical TNBS. We administer TNBS intrarectally in the mice, and we need to perform multiple injections. TNBS will be administered in a suspension of pure ethanol. And in this case, we will also have disruption of the epithelium and thus triggering of the inflammation. And then in the later stages of the disease, we will have fibrosis development. Then I will also show you the TNF model that I mentioned before, which is very interesting, in particular for the studies in Crohn's disease, since we will have a spontaneous development of ileitis rather than colitis as the 2 models I mentioned before. So TNFΔARE mouse basically present a deletion in the TNF alpha gene, which leads to an increased stability of the mRNA of TNF that finally results into an overexpression of the protein. So here the mice will develop ileitis within 7 several weeks, and we will reach a peak of disease severity in between the 20 and the 35th week of the mice. And as we can see here, with H&E, we have thickening of the wall of the gut, and also we can see an increased deposition of collagen in TNF mice compared to the control ones. So then we will move on with the disease severity monitoring. This is not very specific for fibrosis, but it's still something that we have to do to monitor our mice. And so we have parameters that can be used for all the models that I mentioned so far. So the first one will be the disease activity index, which takes into account the body weight loss, stool consistency, and presence of occult blood in the feces. And then also with endoscopy, we are able to monitor the disease severity, but of course in a more invasively way, since the mice has to be exposed to anesthesia. Then we will move on with the most interesting part for us, which is how to assess fibrosis in the mice. So at the moment, the golden standard remain the histological assay. So we have mainly Picrocereus red staining and Masson's trichome. So with Picrocereus red, the advantage is that we can image our slides both in bright field and in polarized light. So with bright field, we will see collagen detection in red, which of course can be then quantified. While with polarized light, we can also further discriminate in between collagen 1 fibrin and collagen 3 fibrin. So collagen 1 will be detected in red or orange, while collagen 3 will be detected with a more yellowish green color. Then we can also perform Masson's trichome staining, where collagen, as my colleague Bogin mentioned, for the human part, will be detected in blue. Then collagen can also be detected with a colorimetric assay. So we have, for example, the circle assay. In this case, we will need to extract the collagen from our sample. This can be applied both to tissues as well as to cells. And then collagen will be stained with a dye, which is generally Picrocereus red, and then it can be quantified with the assay. Then we also have another type of assay that we are using in our lab, which is the type 3 collagen turnover assay, basically, that uses biomarker for collagen degradation, such as C3M, and collagen production, so the ProC3. And basically the ratio between these two parameters show how the turnover of collagen 3 is increased when mice are exposed to DSS compared to the control ones. And this happens actually already at the very early stages of DSS administration. Then we can also use some other biomarkers to evaluate fibrosis and to integrate basically with the previous histological assay or with the collagen quantification, such as collagen 1, alpha muscle, smooth muscle acting, and then TGF-beta that can be assessed with immunohistochemistry and then quantified with qPCR or with Western blot. And then there are also some other biomarkers that have been correlated with fibrosis development, such as the heat shock protein 47, connective tissue growth factor, fibronectin, and synaptophysin. Then another interesting tool that is now becoming let's say, interesting for fibrosis assessment is magnetic resonance imaging, so particular magnetic resonance enterography. So here it's still quite challenging to discriminate between inflammation and fibrosis, so further development of this tool still has to be performed in order to use it as a fibrosis to monitor fibrosis, but there are interesting studies showing how some specific sequences, such as the magnetization transfer ratio, as well as the texture entropy, so texture analysis performed on T2 weighted analysis, can nicely assess and identify fibrosis. So this is something that we're still also trying to perform in our consortium. So hopefully in the next months, we will be able to come up with sequences or with integration of sequences that will allow the use of MRI for detection of fibrosis in mice, which of course will lead to a non-invasive, basically, manner of fibrosis detection. So to To summarize what we said before, the main two, the most commonly used model for fibrosis studies are DSS and TNBS induced colitis, which share disease severity parameters that can be used to monitor the development of colitis. At the moment, still the golden standard technique for fibrosis assessment is the histological assay, so staining for Picrocereus red or Masson stricum. But then there are some other biomarkers that can be used to integrate our data, and hopefully in the next in the early future, we will be able to use non-invasive techniques such as MRI to distinguish and discriminate between inflammation and fibrosis. So thank you.